Welcome to today's lecture on the fascinating field of biochemistry, specifically focusing on the regulation of nucleotide metabolism. Nucleotides are crucial building blocks of DNA and RNA, and the intricate regulation of their synthesis and degradation is paramount for maintaining cellular homeostasis. Today, we will delve into the various mechanisms involved in the control of nucleotide metabolism, exploring topics such as feedback inhibition, allosteric regulation, phosphorylation control, epigenetic regulation, and many more. So, without further ado, let's begin exploring the intricacies of nucleotide metabolism regulation. In nucleotide metabolism, feedback inhibition plays a vital role in maintaining optimal nucleotide concentrations within the cell. This regulatory mechanism ensures that excessive nucleotide production is prevented while also allowing for the synthesis of nucleotides when they are needed. Feedback inhibitors act by binding to key enzymes involved in the nucleotide biosynthesis pathway, thereby allosterically regulating their activity. Examples of feedback inhibitors include purine and pyrimidine nucleotides, which exert negative feedback control over various enzymes in their respective biosynthesis pathways. These feedback inhibition mechanisms fine-tune the overall nucleotide synthesis process and prevent the accumulation of excessive nucleotides. Apart from feedback inhibition, allosteric regulation is another crucial mechanism controlling nucleotide metabolism. Allosteric regulation involves the binding of molecules, known as allosteric effectors, at specific sites on enzymes, resulting in a conformational change that alters the enzyme's activity. In nucleotide metabolism, the allosteric regulation of key enzymes, such as ribonucleotide reductase, ensures precise control over the synthesis of deoxyribonucleotides required for DNA replication and repair. Allosteric effectors, such as ATP and DAP, modulate the activity of ribonucleotide reductase, regulating the balance between the production of deoxyribonucleotides and the availability of their precursors. Phosphorylation control represents another noteworthy regulatory mechanism in nucleotide metabolism. Phosphorylation, the addition of a phosphate group, can activate or inhibit enzymes involved in nucleotide synthesis. For instance, phosphorylation of enzymes such as CAD, carbamoyl phosphate synthetase 2, aspartate transcarbamylase, and dihydroorotate, can activate or inhibit their activity, coordinating nucleotide synthesis with the metabolic needs of the cell. Additionally, phosphorylation events can modulate the activity of enzymes in salvage pathways, which recycle nucleotides from DNA and RNA breakdown, further contributing to the regulation of nucleotide metabolism. In response to cellular stress, such as DNA damage or nutrient deprivation, nucleotide metabolism undergoes significant alterations to ensure the cell's survival. Under stress conditions, cells activate DNA damage response pathways, leading to the upregulation of nucleotide biosynthesis enzymes to facilitate DNA repair. Conversely, nutrient deprivation triggers a different response, resulting in the downregulation of nucleotide biosynthetic enzymes, directing resources to other essential cellular processes. These adaptive mechanisms highlight the intricate interplay between nucleotide metabolism and cellular stress response pathways. Epigenetic modifications, such as DNA methylation and histone modifications, have emerged as important regulators of nucleotide biosynthesis genes. Methylation of cytosine residues within promoter regions of nucleotide biosynthesis genes can silence their expression, limiting nucleotide production. On the other hand, histone modifications, including acetylation and methylation, can either activate or repress the transcription of nucleotide biosynthesis genes, further influencing nucleotide metabolism. The interplay between epigenetic modifications and nucleotide biosynthesis genes highlights the complex regulatory mechanisms involved in maintaining nucleotide homeostasis. Substrate channeling is a fascinating phenomenon observed in nucleotide salvage pathways, where intermediates are physically channeled between enzymes to enhance metabolic efficiency. In nucleotide salvage pathways, enzymes responsible for recycling nucleotides from DNA and RNA breakdown form multi-enzyme complexes, allowing for direct transfer of intermediates without their release into the aqueous environment. This process minimizes wasteful diffusion and shields reactive intermediates from potential degradation, ensuring efficient nucleotide salvage and preventing the unnecessary consumption of cellular resources. Dysregulation of nucleotide metabolism has been implicated in various aspects of cancer biology, including uncontrolled proliferation, genomic instability, and resistance to therapy. Cancer cells frequently exhibit enhanced nucleotide synthesis to support their high proliferative demands. This dysregulation may arise due to mutations in key enzymes, resulting in constitutive activation or loss of regulatory control. Understanding the alterations in nucleotide metabolism in cancer provides valuable insights into novel therapeutic targets that can be exploited to selectively target cancer cells while sparing normal cells. MicroRNAs, miRNAs, small non-coding RNAs, have emerged as important players in the post-transcriptional regulation of nucleotide metabolism enzymes. These miRNAs bind to complementary sequences in the messenger RNA, mRNA, of target genes, leading to their degradation or translational repression. Several miRNAs have been identified that specifically target nucleotide biosynthesis genes, contributing to the fine-tuning of nucleotide metabolism. Dysregulation of miRNA expression can perturb nucleotide homeostasis and contribute to various disease states, underscoring the importance of miRNA-mediated regulation in nucleotide metabolism. The regulation of nucleotide metabolism extends beyond miRNA-mediated post-transcriptional control and encompasses a wide array of transcriptional regulatory mechanisms. Transcription factors can directly bind to the promoters of nucleotide metabolism genes, activating or repressing their expression. Hormones and growth factors can also modulate nucleotide metabolism by influencing the transcriptional activity of key enzymes involved in nucleotide synthesis and degradation. The integration of transcriptional and post-transcriptional regulatory mechanisms enables precise control over nucleotide metabolism in response to varying cellular demands. Hormones exert significant influence over nucleotide metabolism, coordinating cellular processes such as growth, development, and reproduction. For instance, insulin stimulates the synthesis of nucleotides by promoting the transcription of key enzymes involved in nucleotide biosynthesis. On the other hand, glucocorticoids and glucagon can inhibit nucleotide synthesis to redirect cellular resources towards other metabolic pathways during stress or fasting conditions. Hormonal regulation represents an intricate layer of control that ensures nucleotide metabolism is finely tuned to match the metabolic requirements of different physiological states. In addition to nucleotide biosynthesis control, the regulation of nucleotide catabolism is equally crucial in maintaining nucleotide homeostasis. Nucleotide breakdown leads to the generation of nucleosides, phosphate, and bases, which are subject to further degradation. Enzymes involved in nucleotide catabolism are tightly regulated to prevent the accumulation of toxic metabolites and to recycle valuable components for nucleotide synthesis. Various regulatory mechanisms, including feedback inhibition and substrate availability, govern the balance between nucleotide breakdown and synthesis, ensuring the cell's nucleotide pool remains balanced. Nucleotide metabolism is intimately connected with several other metabolic pathways in the cell, allowing for cross-regulation and coordinated control. For example, the pentose phosphate pathway produces ribose 5-phosphate, a key precursor for nucleotide biosynthesis. Similarly, amino acids serve as nitrogen donors for purine and pyrimidine synthesis, illustrating the interdependencies between nucleotide metabolism and amino acid metabolism. The crosstalk between nucleotide metabolism and other metabolic pathways ensures the efficient utilization of available resources while maintaining nucleotide homeostasis. In conclusion, the regulation of nucleotide metabolism is a highly intricate process that involves a multitude of mechanisms. From feedback inhibition and allosteric regulation to phosphorylation control and epigenetic modifications, numerous factors contribute to the fine-tuning of nucleotide synthesis and degradation. Understanding the nuances of nucleotide metabolism regulation is crucial for unraveling the pathophysiology of various diseases, including cancer, and may pave the way for the development of novel therapeutic interventions. As we continue to uncover the complexities of nucleotide metabolism, we gain valuable insights into the fundamental processes that drive cellular function and health.